Now we're going to talk about microbial ecology. And this is a really nice way to finish before the final exam because to talk about microbial ecology, we have to bring in a discussion of all the other concepts that we've covered throughout the term. We have to talk about metabolism, we have to talk about microbial growth, control of microbial growth. We want to talk about microbial mechanisms of pathogenicity. All of these become relevant to understanding microbial ecology. So ecology is the study of, when we talk about traditional ecology, ecology is the study of how different components of a system fit together. So when we're talking about a natural environment, what we could traditionally think of as an ecosystem, we're talking about how the trees fit together with the grasses, fit together with the insects, fit together with the herbivores and carnivores, fit together with the soil, with the seasons. So in ecology, we study how the non-living or abiotic parts of an environment interact with the living components of the environment and how the different living components fit together. Microbial ecology, when we're talking about that, we're talking about how different microbes affect each other, how the environment affects microbes, what about the environment affects microbes, and also how microbes affect their environment in turn. So there are lots of different kinds of microbial ecosystems. There's soil microbiomes, there are ocean, marine microbiomes. Every animal has a microbiome because microbes are ubiquitous. They're everywhere. They're all over everything. We're going to focus on the microbiome of the human body. But as it turns out, the concepts of microbial ecology they can be generalized to pretty much any microbial ecosystem. So some relevant review information for the final exam. Remember back to the chapters on microbial growth and controlling microbial growth. We talked about, so in those chapters, we talked about environmental conditions that affect microbial growth. And here that becomes relevant again. So we talked about the different ways, for example, microbes acquire carbon and energy. We have heterotrophs that build carbon structures in their cells by acquiring carbon building blocks from other organisms, from sugars, from fats, from proteins, from other organisms. We have autotrophs, on the other hand, that acquire carbon from carbon dioxide in the atmosphere, a non-living source. When it comes to energy, we have chemotrophs that acquire energy by breaking down molecules, like sugar, for example, and harvesting the energy that is released by the electrons when the molecules are broken apart. On the other hand, we have phototrophs that harvest energy from photons in sunlight instead of from chemicals. So what are pathogens? Well, we've talked about this earlier in the term, right? There's a reason microbes invade your body. A pathogen invades your body not because they need sunlight. If they needed sunlight, you wouldn't find them inside your gut, right, or in your lungs. There's no sunlight in there. Pathogens are chemoheterotrophs. They are in your body to get energy from the molecules that build your body. They're eating you. They're getting the chemicals from your body and they're getting the energy from your body. So they get carbon building blocks by breaking down your cellular material. They also get their energy from breaking down that same material. In that way, they're much like you. You eat other living things. You eat plants. You eat animals. You eat those things because you are a chemoheterotroph as well. You get both your energy and your carbon building blocks from those other animals. So in that way, pathogens and us are very similar. Now, as it turns out, your mutualists, your good microbes, are also chemoheterotrophs. They also have a reason for being in your body. They are there because it is a good source. Your body is a good source of energy and of carbon. They take that from you. So your mutualists are extraordinarily similar to your pathogens. They have, in terms of lifestyle, they are very much the same. There is only this minor difference in that parasites and pathogens take more from you than they give back. They are not worth the cost to you. Some of them take far more than they're worth. Some of them might take and not give back much of anything or anything at all. Your mutualists take from you as well. That's why they're in you. But they give you something back that's worth it. They're worth what they cost. Okay? So this is about a balanced exchange. A mutualist is worth what it costs you to keep. A pathogen, it costs far more than it's worth. So your mutualists are going to be chemoheterotrophs. Your microbiome members are all going to be chemoheterotrophs, just like your pathogens, just like you. Nutrients and micronutrients, right? So there are other nutrient requirements other than just carbon. We also need nitrogen. We need phosphorus. We need, those kinds of things to build phospholipids, proteins, genetic material. Those molecules are made up of more elements than just carbon. And I want to remind you that micronutrients are often cofactors, which are necessary to activate certain enzymes. Not all microbes have the same oxygen requirements, right? And not all microbes have the same oxygen requirements, and not all microbes have the same oxygen tolerance. There are obligate aerobes, which need oxygen and are not poisoned by it. We have obligate anaerobes, which don't need oxygen and they are poisoned by oxygen. And we have some things in between. We have some mixtures of different kinds of oxygen needs, different kinds of oxygen tolerances, right? And so different microbes are going to have different oxygen conditions that they need to live in. Temperature. Why does temperature matter? Temperature matters because of proteins and the cell membrane, mainly. So when it's too cold, enzymes and other proteins don't work fast enough to sustain the cell. When it's too hot, these proteins denature. When it comes to the cell membrane, when it's too cold, the cell membrane solidifies, and then it can't work properly. The cell membrane has to be dynamic. It has to have this fluid quality to it. It needs to endocytose and exocytose. It needs to be able to move to allow amoeboid movement, like pseudopodia. Things need to be able to move through the cell membrane. It needs to be able to, like proteins need to be able to move from one side of the cell to another. So this cell membrane has to be fluid. It has to have this dynamic quality to it. When it's too cold, those lipids that make up the cell membrane are going to solidify like butter, and it's not going to work properly. And that can cause death for the cell. When it's too hot, it might just dissolve. It could totally disintegrate. The cell membrane has to be in the sweet spot, not too cold, not too hot. It needs to retain some fluidity, but also some structural integrity. When it comes to pH, pH matters because when it's too acidic, proteins denature. When it's too basic, proteins denature. And then tonicity. Tonicity is how much solute is dissolved in water in the external environment. So some microbes are adapted to pure water. Some microbes are adapted to super salty environments. And some microbes are adapted to somewhere in between. Right? So the list goes on. The point is every microbe is adapted to different conditions. And some microbes can actually change the conditions of the environment in which they establish. So remember the slide from way back in the microbial growth chapter? Sometimes a microbe cannot be isolated in the lab without recreating its entire ecosystem because its many environmental requirements are so specific and so complex. That was a list of variables that we understand that we need to consider when trying to grow a microbe in a lab. But there could be other environmental conditions that are not on that list, but that are important to certain microbes. And that might be part of the reason we don't know how to isolate individual needs of individual microbes in the lab. That might be why we can't culture certain microbes in a lab. In which case, sometimes the only solution is like, I don't know exactly what's important, so I'm just going to recreate the whole environment in the lab. Now, when we talked about this list of environmental conditions needed by a microbe, when we listed that before, it was because we were trying to solve the problem of laboratory isolation. How do you isolate these microbes that we know exist based on DNA evidence, but we can't grow them in a lab? What do we need to consider? What are we missing? But another reason it's important to consider a microbe's growth optima for these different environmental conditions is because it can teach you about microbiomes. Why is microbe species A really abundant under these conditions, but really rare under these other environmental conditions? And is that important? Do we want to encourage species A, or do we want to dampen it down? So by understanding optimal growth conditions, we can start to understand how to preserve a healthy microbiome, how to reduce the growth of pathogens and enhance the growth of our good microbes. Your reading talks about microbial antagonism, and this is a term used to describe the different ways microbes antagonize each other. Many microbes acidify their environment, for example. This happened with the first anaerobic bacteria on the planet, and it also happens in our bodies. For example, there are many types of fermentation, most of which create acidic waste products. One example we've already discussed is how lactobacilli maintain an acidic environment in either yogurt cultures, bread cultures, or the vaginal environment. If the pH changes, other normal vaginal microbes can grow unchecked. The microbes most commonly associated with bacterial vaginosis have been shown to actively increase the pH of whatever environment they inhabit. In other words, once the pathogenic bacteria establish, they contribute to altering the environment to be more favorable to themselves. Lactobacilli like it acidic, and they make it acidic. If you remove the lactobacilli and you increase the pH so that it's more neutral, these other bacteria that we don't like overgrowing, they start overgrowing and they start neutralizing the environment even further. They raise the pH even further so that it's less favorable to lactobacilli and more favorable to themselves. That antagonism between lactobacilli and the other inhabitants of the vaginal environment is called microbial antagonism. Both of them are antagonizing each other. Now, we like microbial antagonism when lactobacilli is dampening down the bacteria that end up causing BV. We don't like it when the BV microbes antagonize the lactobacilli. So it's not that microbial antagonism is always good for us or always bad for us. It's just a description of this dynamic between different microbes as they compete with each other for a foothold in your human body, which is their ecosystem. We talked about how microbes can change the temperature of their environment by burning calories in, for example, a compost pile. So remember that you kill pathogens using heat generated by microbes themselves to make compost that is safe to grow human food in. This is a similar concept to pasteurization. If you are using this compost to grow veggies that humans are going to eat, it's really important to kill off all the human pathogens in the compost pile. You don't need to sterilize the compost, you just need to remove the human pathogens. And you do this by letting the mesophiles, which like moderate temperatures, grow in the compost, metabolize in the compost, burn calories, and generate enough heat that they eventually kill themselves off because it's too hot. They've made it too hot for themselves. Mesophilic bacteria generate heat as they metabolize, just like you do when you exercise. All the heat of that mesophil metabolic activity eventually makes it too hot for the mesophils. The mesophil enzymes denature, the mesophil cell membranes melt, and they die. But thermophiles are now quite happy in the new hot environment, and they continue metabolizing and generating heat, keeping it too hot for the mesophiles, which are going to be the human pathogens, because your human body is perfect for a mesophil. It's a moderate temperature. So when you kill off all the mesophils, you haven't killed off all the bacteria, right? It's not sterilized. The thermophiles are still there, keeping it hot. But you've killed off anything that you need to worry about because it's going to be the mesophiles that want to infect you. The thermophiles aren't going to really like your body because it's too cool for them. Microbes can also alter temperature in your body by interacting with your immune system. And this can have effects on microbial community composition. So when your temperature rises, it alters the ratio of different species of microbe in your microbiome. Because some elements of, yes, the purpose of fever is to kill off pathogens. But in your microbiome, some species of your good microbes are going to be more tolerant to your fever than others. And that's going to alter the ratios between species. On a related note, microbes compete for carbon, nutrients, and space. Some microbes are better at this than others. But importantly, every microbe is better at competing when they are living in their ideal environmental conditions. So if oxygen concentration is just a little too high or pH is just a little too low for a particular microbe, it might not die, but it will not compete as well with the other microbes. And then if the environmental conditions change, a different species of microbe might be favored. So when you change the environmental conditions of your body, you might not be extinguishing certain species from your body. You might just be changing the ratios between species. Species A really likes it when your body is really acidic. Species B likes it when it's more neutral. They might both exist under both conditions, but who's dominant is going to be what's affected. So to explain what I mean, consider this chart showing optimal pH values for 3 different microbes. You have pH on the x-axis, so over here near 0, that's acidic. Over here near 14, that's going to be basic, right? And then you have cell microbial growth. This is cell growth or microbial growth on the y-axis. Okay. You can see that at pH 7, right here in the middle, all 3 of these species can grow. So here in blue, I should just reiterate, here in blue, species in blue is an acidophile. This species likes it acidic. It prefers acidic environments. In orange, you have a neutrophile. This microbial species really prefers a neutral pH. And then in green here, you have an achilophile. The achilophile really prefers a basic pH or an alkaline pH. Right? Okay, so at pH 7, all 3 species are growing. So you have some of the species in green, some of the achilophile, some of the acidophile, and a lot of the neutrophile. So all 3 are inhabiting the same environment, but only one is growing really, really well. This species, the neutrophile, is going to be the dominant species at a neutral pH, but the acidophile and the achilophile might still be there, but just in lower numbers because they don't like it as much. They can't compete very well because they're not in their optimal environment. They're weakened by having a lack of optimal environment for them. Now, if this environment changes to a pH of 4 down here, the yellow microbe will still survive, the neutrophile will still survive, but it's going to be more rare. So like maybe pH of 4, that's probably right here. So the neutrophile is still there, but it's no longer the dominant species. The acidophile is now the dominant species because now we're in an acidic environment. It's still not its optimal environment, but it's far more optimal relative to the other 2 species. So the acidophile starts growing better, the neutrophile stops growing as well, and the achilophile, in this case, it's still just barely hanging on, but it is pretty rare now. So you still have all 3 species, but you have a shift in ratios. The dominant species has changed. Okay? So at a pH of 7, the neutrophile was dominant, and the achilophile and the acidophile were equal. They were of equal abundance, and they're both fairly rare. At a pH of 4, the acidophile is the dominant species, the neutrophile is a bit rare, and the achilophile is extraordinarily rare. Okay? So you have a different and relative abundance between these species. All right, so this phenomenon where you have, when the environment shifts, you have a shift in abundance between species. Sometimes you'll have an extinction of a species, but sometimes you merely have a shift in dominance. And that phenomenon is true for any environmental condition. This could be a graph about different oxygen concentrations, different salt concentrations, different temperatures. So tolerance for oxygen, salt, pH, temperature may overlap between species, but different conditions are going to favor different species most, and so are going to change the relative abundances between species. Microbes cooperate with each other too. So microbiomes are full of antagonistic interactions between species. You have microbial antagonism constantly, where species A is antagonizing species B to try to get a foothold. Right? But microbes also cooperate with each other. Remember that one of the reasons most microbes have not been isolated in culture is because they cannot live without a close association with a partner microbe. Right? So remember this slide from way back when I talked about how some bacteria cannot grow in isolation but require a partnership with another microbe for survival. Culturing 2 or more species together can actually help solve some of the mysteries of laboratory cultivation, laboratory isolation of microbes. And this can be for a number of reasons. Some microbes are really good at preserving the environmental conditions required by their neighbors, such as the correct pH. Some microbes live off of very specific waste products produced by another microbial species. Some microbes produce the enzymes to digest certain foods in the environment, and another microbe mooches off the enzymes that a different species makes. Okay. So let's look at a commonplace example. One example would be yeast and lactobacilli in sourdough bread cultures. Okay, so in sourdough culture, you have you can grow it in your fridge. You put water and flour in a kind of like yogurty, mix it to a yogurt consistency, and you freshen up the flour every day. You throw some out, add some fresh, and you just keep it, and eventually you get a sourdough culture. You get this like all of these microbes will colonize it first, and it'll be gross and disgusting, and then eventually this balance will be struck between mainly yeast and lactobacilli. So the partnership between lactobacilli and yeast in a sourdough culture is really cool. So what this lactobacilli ferment sugars. They ferment and they produce lactic acid. That's responsible for the sour taste of sourdough. And remember, fermentation is a partial oxidation of sugar. So when you're fermenting sugar, you're only breaking it down part of the way, versus aerobic respiration, where you completely strip all of the electrons off and release all the carbon as carbon dioxide. Fermentation isn't that. It's only a partial oxidation of sugar. It's only partially broken down. Okay, so the yeast it's not very good at eating the sugar, the full sugar, but it really likes to digest the pre-digested fermented sugar, the waste products from lactobacilli fermentation. So yeast loves the partially digested sugar, the lactic acid, the waste product produced by lactobacilli. This is sort of like one man's trash is another man's treasure, right? The waste product of one is the foodstuff of the next. And this kind of exchange happens constantly in your body, in your microbiome. So, microbial cooperation. One microbe might preserve the environmental conditions necessary for other microbes to survive. So maybe one microbe is really critical for preserving the optimal environment for a particular community. One microbe's trash is another microbe's treasure. Waste product of one can feed another. But microbes might provide food for each other in other ways, by being food for another microbe. And actually, I'm not sure if that belongs in the cooperation or antagonism slides, but this is a food chain. You have food webs in your microbiome where some microbes are the prey of other microbes. And so the predator relies upon the prey for survival, and that can be another way two microbes rely on each other. Waste products are often toxic to the microorganisms that produce them. So waste removal by other organisms not only feeds the one that's eating the waste, but it also detoxifies the environment of the microbe that produced the waste. So it's sort of like, when one man's trash is another man's treasure, that's kind of great because the trash is taken away from the one who doesn't want it, and it's usable by the one that does want it. Some microbes don't have the genes to live in certain adverse conditions unless they are infected by a prophage. So viruses are microbes too. And it turns out, we talked a lot about how viruses can infect bacteria and give them new properties. And often we thought, that's dangerous for the bacteria because when a prophage is in its lysogenic cycle, it's living silently inside the bacteria and passed through generations of bacteria, but at any moment it can pop out of the bacteria and kill it if it enters the lytic cycle. So we thought of the lysogenic cycle as bad, but actually sometimes it's good for the bacteria. It all depends on what kinds of genes the prophage is bringing. So often a prophage can bring tolerance genes to a bacteria. Like maybe the prophage brought antibiotic resistance genes. Maybe it brought resistance to salty environments. And so now that microbe can tolerate a more saline environment than it used to be able to tolerate. So one concrete example is in hot springs. Certain bacteria, so in a hot spring, it's like boiling temperature. And there are bacteria and archaea that live in there. There are bacteria that live in hot springs that actually cannot tolerate the high temperatures of the hot spring unless they are infected by a virus. The viral prophage inserts a temperature-tolerant gene into the bacterial genome, and that allows that strain of that bacterial species to live inside the hot spring. And other examples would be some microbes do not produce necessary defense chemicals for warfare with other species unless they're infected by the virus. The virus might bring them the ability to create chemical weapons through genes. There is some evidence that viruses might have important roles in our microbiome, but this is even, if you want to look up the human virome, microbiome is usually used to describe bacteria, even though it technically also includes protists and fungi and maybe some archaea. But the human virome includes all the viruses that live in your body. Those include animal viruses that infect our human cells, but it also includes all the bacteriophages that infect and affect your microbiome. And it's really interesting to think about, because a lot of those bacteria would be completely different if it weren't for their prophages. So it's really hard for us to parse out right now. We know very little about the virome, but it's almost certain that the virome is an important and influential factor in how our microbiome treats us. For example, bringing this back to bacterial vaginosis. Some women get bacterial vaginosis repeatedly, over and over and over again. And it's really a mystery why. Because, again, bacterial vaginosis is not caused by a single species of bacteria. It's not a pathogen. It's dysbiosis. So it's not that when the woman has BV, she gets the same pathogen over and over again. It's just that she gets bacterial overgrowth of her normal vaginal microbes over and over again. And we don't know why. Her lactobacilli is not creating the balance of the vaginal microbiome that it's supposed to. Why is that true for some women? Well, it turns out BV might recur in some women continuously because there's this virus that kills lactobacilli. Now, the virus that kills lactobacilli is really important in the yogurt industry because lactobacilli also creates yogurt for us. So this virus that demolishes lactobacilli, it demolishes yogurt cultures. If that same virus can also infect the lactobacilli that is responsible for vaginal health, it would explain why BV, bacterial vaginosis, cannot be combated with antibiotics, because antibiotics don't affect viruses. So that is also really interesting to think about. That's a level of infection that we are not used to thinking about as humans. There could be a virus that destroys our good bacteria that protects us against bad bacteria and something like bacterial vaginosis, dysbiosis. So the point is, it is not only through our immune system that our microbiome helps us fight pathogens. So yes, our microbiome trains our immune system so that it functions properly and functions strongly. But also members of our microbiome control each other's growth by competing with each other and also by excreting chemicals. They also fight off pathogens in the same way. They are considered to be elements of our innate immune response. Pathogens that enter our body have to get past our good microbes before they can infect us, and that is a difficult task, as it turns out. So you can see how an understanding of ecology, as complex as ecology can be, is extremely relevant these days to understanding human health and human diseases. And this brings us full circle back to the very first chapter we covered this summer, in which we talked about the development of germ theory. Prior to germ theory, there were various superstitions to explain disease, one being demons and sin. But experiments conducted by many scientists helped develop germ theory over a couple of centuries. Some of the people we discussed were Robert Hooke, Anton van Leeuwenhoek, Joseph Lister, Robert Snow, Louis Pasteur, Robert Koch. And just to remind you who each of these were, Robert Hooke saw the first cells, the first, not microbial cells, but the first cells, plant cells, in his new microscopes. Anton van Leeuwenhoek discovered the microbial world with his 400 plus microscopes. Joseph Lister was a surgeon who initiated the use of phenol for sterile technique during surgery. Robert Snow was perhaps the first epidemiologist who tracked, even before germ theory was a thing, he tracked the source of cholera to an infected well when there was a cholera outbreak. Louis Pasteur made major contributions in the golden age of microbiology, including pasteurization, discovering that microbes can cause disease, discovering that microbes can create wine, discovering that microbes destroy wine. Right? So, and he also accidentally stumbled upon vaccines. And we talked about Robert Koch, who created Koch's postulates. Right? And Koch's postulates, the point of them is to identify the microbial cause of a disease in a consistent manner. And this is where the microbiome is where things get really fuzzy when it comes to Koch's postulates, right? So in Koch's postulates, according to Koch's postulates, for a microbe to be considered the source of a disease, it must be found in abundance in all organisms that suffer from the disease, but it should not be found in healthy organisms. Also, the microbe must be able to be isolated from a diseased organism and grown in pure culture. The cultured microbe should cause disease when it's introduced into a new healthy animal, and the microbe must be re-isolated from the inoculated, diseased experimental animal and identified as being identical to the original cause of disease from step 1, right, or step 2. And remember, Robert Koch did this initially with cows and anthrax. So there was a cow sick with anthrax. He drew some blood, found a microbe in it, isolated the microbe in the lab, infected a healthy cow with that microbe. That cow also died of anthrax. He looked at that dead cow's blood, found the exact same microbe as the one that he initially isolated from the original sick animal. Now, these were really, really useful in the first golden age of microbiology because before Koch's postulates, before germ theory, we didn't even know that there were microbes. And when we did know that there were microbes, we didn't know that they could cause disease. We thought that demons caused disease and karma and things like that. So the idea that this thing that's so small that you can't see it could kill an entire village, that was crazy. And so Koch's postulates really helped us in the beginning show to the general public that yes, there are these tiny organisms, and yes, they cause disease, and this is how they do it. But now we are finding that other diseases, which we did not originally believe to have a microbial cause, such as obesity, irritable bowel diseases, asthma, allergies, depression, even arthritis and MS, are in fact implicated by microbes, but not in ways which fit into these postulates. We talked before about how some pathogens cannot be identified using these postulates because some cannot yet be isolated in pure culture. But the diseases I just listed, though microbially induced, are not caused by a particular pathogen. They might be caused by too few microbes, too many microbes, an imbalance in microbes, or by a lack of a specific kind of microbe. So lack of microbes, too many normal microbes, the improper balance between normal microbes, all of those things, that's not a pathogen causing disease. That's not having the right ratios or the right kind of microbe. Okay? And then still other diseases like meningitis, bacterial vaginosis, Clostridium difficile, C. diff, and botulism are caused by pathogens more or less, but still cannot be determined to be pathogens using these postulates. They break the mold. For example, remember that with C. diff infection, with Clostridium difficile, which you'll read about for your homework, it's not a disease that can be transferred from one person to another because everyone has C. diff. And no matter the level of exposure, it will remain a rare component of a healthy gut microbiome. You can get infected by another person's C. diff all you want. If you are a healthy person, it's not going to do anything to you because you already have it. It's only by removing all the other microbes that keep C. diff in check through microbial antagonism that C. diff can then overtake the colon and cause problems. The gut environment is not optimal for C. diff. C. diff can live in the gut, but it's going to be rare. It's not going to be competing well with all the other microbes in your gut. All the other microbes in your gut, they hog all of the nutrients, all of the space, and C. diff just barely hangs on in a normal, healthy adult gut. But if you take an antibiotic, you remove all the good microbes, and then you add in C. diff, well, now there's nothing blocking its growth. It may not be its optimal environment, but there's nothing else around keeping its growth in check. Okay? And this is why antibiotics often don't work for C. diff infections, because the infection is made stronger when none of the good microbes are around. So in a case like C. diff infection, postulates 1 and 2 are still good. The microorganism is found in abundance in all organisms suffering from the disease. It's found in healthy organisms, but not in very large abundance, so that one's maybe a stretch. And then the microbe can be isolated in pure culture. But 3 is not good because it's never going to cause disease if introduced into a healthy person, right? Because healthy people already have it. It's only unhealthy people who've taken antibiotics who are at risk. So if we were face-to-face, I would do an activity now with you where you got to read about all these pathogens and diseases that are not caused by a traditional pathogen, but rather by pathogens that break the mold of Koch's postulates. And it turns out there's quite a few of them. I'm going to make a video in which I'll describe some of these microbes, and I'll let you read, you'll have access to them. It's totally optional now for this online class, but it's really interesting because you can read about cervical cancer. You can read about irritable bowel diseases. So in the case of irritable bowel diseases, just to make a plug for how interesting this material is, when it comes to irritable bowel diseases, you can resolve the symptoms by adding a parasite. Usually we think of parasites as bad, but for someone with chronic inflammation, adding a parasite might be good. And there's other, strep throat and meningitis, these things we think of as, just caused by pure pathogens. It turns out the microbes that cause those diseases are found in everyone. And we actually don't really understand very well why those pathogens cause disease in some people but not others. So if you have the time, I recommend, at least downloading these articles for later because they really are interesting cases and they really exemplify how much more complicated microbes and pathogens, even just pathogens, are than we traditionally thought. The rest of this, I have so many videos on the microbiome which are super interesting that I would have loved to show you in class. So I'm going to post them all on the website, and I really hope you take a look at them. Especially, there's one by Rob Knight. Rob Knight works at UC Boulder, and he is perhaps the premier world researcher on human microbiome. He created the software, the analytical tools, the analytical methods for learning about about any microbiome anywhere. But his research is especially interesting to us as humans when we look at his research on the human microbiome. And he has this phenomenal TED Talk that talks about a lot of the things in this lecture, but he has different, of course, his own way of describing it. So I hope that you'll look at some of these videos because they're really fascinating.